Okay. Hi, everyone. So, my research paper was titled Undercover Agents, Blood Doping and Its Detection in Endurance Sports. So, starting off with my PICO question, I had to modify it a couple times to really figure out exactly I wanted to research. So, originally, I started off with what effect does blood doping have on elite level college athletes within the ranges of 18 to 25? And there really wasn't much research to find on this kind of very specific topic. So I decided to kind of modify it to get a more well-rounded search, and ended up changing it to what are the effects of blood doping and strategies to detect it within athletes. Okay, so a little introduction on blood doping. Blood doping is defined as the act of improving performance by boosting the blood's ability to carry an increased amount of oxygen. So this is basically the reason that blood doping has become such a popular form of cheating for endurance athletes, such as marathon runners and cyclists. This form of blood doping is a subcategory for gene doping, which is enhancing or engineering an individual's genetic makeup for performance enhancement. The act of blood doping can increase hemoglobin and erythropoietin levels. These are substances that carry oxygen in the blood and produce red blood cells, respectively. It's these substances that make blood doping so appealing for endurance athletes, right? So, an increase in hemoglobin and erythropoietin can increase the ceiling for an athlete's aerobic capacity. So, corporations such as WADA, which is a World Anti-Doping Agency, and the International Olympic Committee, or the IOC, have begun to crack down on blood doping. So when it actually comes down to the process, um, there's a couple different ways athletes can blood dope. So the most popular form is blood transfusions. These can date back several decades, and it's one of the original forms of blood doping. So this process is done by infusing the athlete with either their own blood or blood of a donor from the same blood type um, as a transfusion. So autologous blood doping is a form of blood transfusion where the patients use their own blood. So this form is harder to detect, but is a little bit less effective in increasing red blood cell levels. And then there's also homologous transfusions, and these use blood from a donor. These can be done one in between 1 and 7 days pre-competition. However, this form is easier to detect through drug tests because they're not using their own blood, obviously, and it can show different signs as far as tests go that can kind of lead these anti-doping agencies to realize that they have been doping. So, like I said, this form is easier to detect through drug tests, and it also puts patients at risk for infections and different reactions their bodies can have. Another form of blood doping is used by blood substitutes such as hemoglobin-based oxygen carriers and perfluorocarbon. These are agents that bind and deliver oxygen to the tissue. They're originally used for patients with large amounts of blood loss. They do show potential for increasing like aerobic capacity and performance enhancement, but they haven't really been tested quite as much as like blood transfusions, and they're a little more rare and not very well documented. So, blood doping also has been around for decades, but uh, testing and detection still remains a pretty large issue with anti-doping agencies. So they're constantly testing and forming new ways to detect all forms of blood doping, whether it's transfusions or by erythropoietin or the hemoglobin. So tests can be done directly by studying the genetic side or indirectly by studying the effects of doping throughout the organism. So a new kind of form that these agencies are trying to study is It's called the Athlete's Biological Passport, and this is kind of a groundbreaking thing. So basically, if you're involved in high levels of competition for endurance sports, such as cycling and marathon running, they're starting to compile data on your actual blood work and keep stores of it so they can track levels like at a baseline, and then see during the season and after events if they suspect anything, they can check different levels of like hemoglobin, red blood cell counts, and determine if they think the athlete has been using a performance enhancing drug. So, and then also, like I said, although this can have huge benefits on sports performance, can also leave the athlete at very high risk for health issues. So the use of EPO can be very beneficial in treating health conditions such as anemia within the kidneys, but it can be very dangerous, say just for an athlete that's looking to use it for performance enhancements. All right, so when it came down to conducting my research for my methods, I used two different databases. I used PubMed and then EBSCOhost through the library, and I used 4 different terms. The terms were blood doping and sport, blood doping and detection, blood doping and effects, and then finally blood doping and endurance. And then these were both typed into both databases, and a total number of results were given to me. So for the most part, PubMed gave me a little more resources, a little more articles to review. And then duplicate articles were pulled out between the two. I found 97 total for blood doping and sport, 41 for blood doping and detection, 61 for blood doping and effects, and 19 for blood doping and endurance. So as far as finding the results, the windows were narrowed to fit the years between 2010 and 2021. However, a couple of the sources I found, I think when I typed the searches in, forgot to set some parameters. So a couple of the sources I found were actually from 2007 and 2008, which are still relatively close to the window. And I evaluated those and determined to keep them in the research because they actually gave pretty good findings and results on the topics. So as far as the results themselves, they were randomized control trials, clinical trials, and meta-analysis articles. And I also limited down to full text written in English and primarily endurance athlete subjects. Okay, so now on to the results. I'm not going to break this down too much and overcomplicate things because a lot of the results were very specific and had to deal a lot with the data that was pulled from the searches. But I'll go over a couple of them and kind of explain what they mean and what they led to. My main focus for the results was on detection strategies. So on one of the topics that I researched, they did a study with 21 total participants. 11 of them they did blood transfusions on, and the other 10 were a control group and were not on any sort of performance enhancing drugs. So they were divided into a dope group and a control group. And then the subjects all underwent a full cycling season, and their total hemoglobin levels were tested 10 times throughout the season. And the outcomes from this study basically showed that there was issues with it because of its ability of the test to detect abnormalities of autologous blood transfusions. So it had a difficult time really determining if athletes were doping or not. And then another study that I found kind of interesting with the detection had 34 total participants in it. 14 were endurance trained runners that lived at sea level, and 20 were endurance runners living 2,100 meters above sea level, so at a pretty high altitude. And these sea level athletes moved to a higher altitude and drew blood samples before and during their altitude training. And the high altitude athletes injected erythropoietin every second day for a 4-week period. And the outcomes for this study were actually pretty interesting. So 9 of the 14 sea level athletes showed abnormalities in hemoglobin fluctuations, which those were the athletes that did not participate in the blood doping. So their hemoglobin numbers actually showed abnormalities as if they possibly had been blood doping. And all the EPO injected athletes were detected as expected. So that really didn't come to much a surprise. But those were a couple of the searches that I found and data that was pretty interesting. All right, so kind of discussing all my findings and everything from the research, basically determine, that there is multiple ways to figure out whether an athlete has been blood doping or not. I was able to figure out that there is multiple ways to detect actually if athletes have been doping. A couple of the different articles that I found, detection strategies included urine samples, testing hemoglobin levels and concentrations, and kind of the big thing that came up as far as the future and where testing is going to would be that the athlete's biological passport that I discussed a little bit earlier. This basically, like I said, just gives the agencies such as the WADA, the IOC, kind of a baseline as to where their athletes stand kind of before competition and during competition with all their hemoglobin levels and numbers such as that they can use to determine whether an athlete's been doping. And actually this picture down here in the bottom right corner actually didn't come from any of the articles that I researched, but I found it just online through a Google search, but actually does kind of a good job explaining how this process works. So basically on the top here, it has hemoglobin concentration and blood volume. As those increase, the hemoglobin mass increases, which then leads to an increase in aerobic capacity. Max heart rate and max stroke volume lead to max cardiac output and also an increase in aerobic capacity. That really wasn't discussed too much in my research, but that's another way that it could work. And lastly, max O2 extraction can come from O2 affinity of the blood, which also leads to an increase in aerobic capacity. And then finally here on conclusions, this is still a relatively new method of like performance enhancing drugs for athletes. Detection strategies are still continuing to progress and become better and more efficient at catching professional athletes that are using these forms of cheating, essentially. There's been multiple methods that have been studied and analyzed to detect blood doping, as we just discussed on the last slide, such as urine samples or just through blood work. And the athlete's biological passport will continue to be a very important detection strategy for these agencies if they can, kind of gather as much information as they can and figure out how to pinpoint these tests to make them as efficient as possible and kind of foolproof. Going through the research, there's plenty of ways to really detect it, but pretty much all the methods still had flaws, and there were still instances where people were not blood doping and could still come back as positive or show changes in their blood work just from various other reasons. So basically the research just must continue to grow and develop to where this isn't as hard of a thing to deal with for these agencies. And lastly, here's just a quick little reference slide. So All right. Thank you for listening and have a great day.